Results of meiosis. So what you end up getting is 4 haploid cells. They are gametes, either egg or sperm, that are formed. So you get 4 haploid cells with 1 copy of each chromosome. So you don't have pairs anymore. 1 allele of each gene is present. They're not in pairs anymore. And you have different combinations of alleles for different genes present along the chromosome. So look, here's a combination of genes that's never been around before. This part came from your dad, that part from your mom, and you're giving your baby, to be possibly, a brand new combination of genes. So here's an animation that we should look at. And so you're starting out with an organism, in this case, where 2n equals 4. And so there are the 4 chromosomes. They just crossed over. Now that's meiosis 1. This is meiosis 2. And here you have haploid with n equals 2. So starting again, there's crossing over. There's separating out. That's meiosis 1. Here's meiosis 2. And you get the haploid cells. Goes too fast, doesn't it? Okay, here's another overview of it all. So here's meiosis 1, and you've got 2 very important things happening. First of all, you get crossover, and the second thing that happens is independent assortment, where they line up sort of, this one will line up independently of this one, which will line up independently of this one. And then they'll separate out in anaphase. So that's meiosis 1, and you're separating out homologous pairs that are still duplicated. And then this is meiosis 2. And so you start out with these 2 cells from meiosis 1. They're already haploid, but they are not paired anymore by the time you get here. So here's the beginning of meiosis 2. You have haploid cells, but the chromosomes you can see here are still duplicated. So that's prophase. Metaphase is when they line up in the middle, and anaphase they separate. And what you're separating this time are sister chromatids, so they're no longer duplicated by the time you get to the end. So here's telophase in the beginning of cytokinesis, and you can see you've got 1, 2, it's hard to count them in here, 1, 2, 3 chromosomes. That one's kind of a long one, I guess. But they're single, and they're unduplicated, and this one doesn't have a homologous pair, and that one doesn't have a pair either, and that one doesn't have a match either. That's a homologous one. They will meet up with another one when fertilization happens. Quiz time. Homologous chromosomes join together in a tetrad and may exchange alleles. This is called crossing over. Homologous chromosomes can line up in any order as long as one is pulled to one daughter cell and its homologue is pulled to the other daughter cell. This is called independent assortment. All right, the big picture. What 3 things increase genetic diversity in sexually reproducing organisms compared to asexually reproducing organisms? So this would be a really great essay question for you, don't you think? So there are 3 things. One is that you've got 2 parents. Another is that you've got crossing over to give you new gene combinations. And finally, independent assortment also gives you new gene combinations. So I would want you to state each of them and describe how each gives you more genetic variation. See, it says it here even. What a great essay question that would be. Could be worth lots of points. And then when we do the evolution unit, you can talk about why that variation is so important. So you know this question is about evolution. Why does natural selection favor sex when the going gets tough? In other words, why is there an advantage for offspring that are different from their parents and from each other when the environment changes? And the answer is, if the offspring are different, then hopefully a few will survive the new environment. Meiosis makes gametes, so it's sometimes called gametogenesis, meaning to make gametes. And if it's in males, it's called spermatogenesis. That means making sperm. And if it's in females, it's oogenesis, making eggs. So spermatogenesis makes sperm, and it occurs in the testes. And there are 2 divisions that produce 4 spermatids, and the spermatids will mature into sperm. So spermatids mature into sperm. And men produce about 250 million, let's see, thousand, yeah, that's million sperm per day. Holy cow. Spermatogenesis. So if your 2n happens to equal 4, then you're going to end up making sperm that have 2, n equals 2. 2n equals 4. So notice that you're already haploid here. So this is diploid. These have the homologous pairs have separated out. So these are haploid. These are also haploid, and the difference between them is that these are unduplicated chromosomes and those are duplicated chromosomes. So spermatogenesis ends up making 4 sperm for each cell that you start with. Oogenesis is a little bit different. This occurs in the ovaries of females. And remember that eggs must be really big because they have all that cellular machinery needed to start life. So we talked about that a lot in an earlier unit. And here's what the crazy, crazy thing that happens. You've got to separate out your DNA so that the egg only has a haploid number of chromosomes and the DNA is unduplicated. So that's got to happen. And that does happen. The DNA gets separated evenly. The crazy thing about oogenesis is that cytokinesis is different. So cytokinesis is uneven. So the DNA gets broken up perfectly, but the cytoplasm doesn't. So this ends up giving you a really big cell and an itty bitty tiny cell. And then when this divides, it divides unevenly again. The cytokinesis divides unevenly. So you get a huge cell and a tiny cell. These cells are called polar bodies. If they're fertilized, nothing can happen because there's not enough cellular machinery, not enough enzymes, not enough ribosomes, not enough all that stuff. And even if these get fertilized, they die. If this one gets fertilized, though, it should develop into a zygote and an embryo and a fetus and so on. So the important thing for oogenesis to understand is 2 things: that DNA is separated perfectly and exactly. Whoa. And cytokinesis, that's the uneven one. Everything's not separated out evenly. So 2 divisions produce 3 polar bodies that die and 1 egg. The polar bodies die because of unequal division of the cytoplasm. An immature egg is called an oocyte. Starting at puberty, about 1 oocyte matures into an ovum every 28 days. That's a little bit different from the 250 million sperm a day. So anyway, spermatogenesis, you get 4 sperm, and they're all about the same. And with oogenesis, you make 4 cells, but 3 of them are junk. And that's it.